BioScope Resolve has had a lot of improvements on the hardware in the fact that we've really decreased the noise on the system in both the X, Y, and the Z so that we can get really nice high resolution images. We're able to look at DNA double helix, bacteria rhodopsin, so down in the nanometer range. So that's one big improvement. Another area of improvement is with our peak force tapping, peak force Q&M technology. With the BioScope Resolve, a lot of people are interested in looking at cell mechanics in terms of cell mechanobiology, for looking at how forces affect cells, affect disease states. So we've really improved our peak force tapping. We have a brand new probe, AFM probe, that has a 17 micron tall tip, which is really nice for imaging large topography cells. We also have a new background subtraction algorithm that allows, when you're going over the large topography of the cells, it really allows to get that peak force that you're using really nicely, so it improves the feedback signal, so the tracking is much better. One of the real key things we wanted to make sure the BioScope Resolve is that it applies to various biological research. So everything from molecular biology up to cell biology. So users can get really high resolution images of the structure of various biomolecules like DNA. You can see the DNA double helix, major minor grooves, which are about 1.2, 2.2 nanometers, so very small structures. Bacteria rhodopsin, so looking at the arrangements of membrane proteins, lipid bilayers, how proteins interact with that, as well as looking all the way up to cells, which is not so easy on an AFM. That may sound like small things, but when you're looking at a 2 nanometer molecule versus a 30 micron cell, it takes a lot of that dynamic range to make an AFM to suit going from one end to the other. Mechanobiology and biomechanics is really an emerging field where researchers are really finding that forces play a really large role in how cells react to and behave in their environment. There's a lot of research going on looking at cancer, for example, where they're seeing that cancerous cells are much softer than healthy cells. And one train of thought on that is that when cancer cells metastasize and move throughout the body, they need to be softer in order to kind of move through tissues and things like that. So in that sense, AFM is being looked at as a potential diagnostic tool. Other things are, looking at adhesion measurements. So again, when you're trying to image cells, it's really hard to see single molecules on the surface of these cells. Even with optical microscopy, trying to get down to that resolution in nanometer is still very difficult. So what users have done on the AFM is they've functionalized probes, and then they can use that probe to image across the surface, and they functionalized it with a ligand or a molecule that binds to a specific molecule of interest or a cell surface receptor. And as they're imaging over, they see binding events, and these binding events are recognized as adhesion events, and you kind of get a map of molecular recognition. So you will see how the molecules are distributed over the surface of the cells. And we actually have researchers who are doing this, and they've been able to achieve a lateral resolution of about 4 nanometers in determining the position of these molecules.